Hello, and welcome to part 3 of our lecture series on the endocrine system. And in part 3, we're going to take a look at the pineal and the thyroid glands. Now, the pineal gland or the pineal body is essentially a hormonal secreting structure located in the roof of the diencephalon. So it's essentially attached by a stalk to the roof of the diencephalon. If we take a look at it, we've got two types of cells that are going to be present. We're going to have pinealocytes, which are going to have large irregular nuclei and a basophilic cytoplasm. And the pinealocytes are going to be involved with secreting melatonin, which is involved with a variety of lighting and circadian rhythms. Scattered among these basophilic cells are going to be astroglial cells, and these are glial supportive cells, kind of surrounding and supporting our pinealocytes. We can identify the pineal body by the presence of essentially calcified globular structures. They're staying relatively basophilic. They're called either brain sand or corpora arenacea. So essentially you've got these kind of purplish clusters of calcified material surrounded by these basophilic cells. The presence of the brain sand and its location within the roof of the diencephalon makes it a useful landmark for radiologists. And again, the secretions from this are melatonin involved with regulating circadian rhythms, also helping with like sleep and wake cycles. The next hormone secreting gland that we're going to take a look at within our endocrine system is the thyroid gland. If we take a look at the thyroid gland, what we're going to see is the functional unit are going to be thyroid follicles, similar to what we've got on the slide to the right. So we have these circular aggregates of cells, and we're going to have thyroid follicular cells, which are going to be involved with essentially synthesizing and secreting the peptide thyroid hormones. And so what we're going to have are these kind of spheric structures. We're going to have colloid to the center, kind of that pink, even pink staining appearance, which is going to fill the central lumen, and we're going to have the thyroid follicular cells that are going to be wrapped around it completely. Depending upon the height of the cell, you get an indication of how active this thyroid follicle is going to be. The shorter the cell, the more inactive. So a simple squamous epithelium is going to indicate an inactive thyroid, or at least an inactive thyroid follicle, where a more cuboidal or columnar epithelial cell is going to indicate that it's a stimulated cell and it's an active follicle. Now, these thyroid follicular cells are going to be involved with both the synthesis and secretion of thyroid hormone, thyroglobulin. What's interesting about this is that these thyroid follicular cells are going to be storing the intermediate product extracellularly. They're going to be storing it within the colloid in that central lumen. So if we take a look at this, again, from the production standpoint of the thyroglobulin, what we're going to see is that that thyroid follicular cell is going to be doing what we'd expect of a cell that's producing a peptide hormone. So we're going to see synthesis and storage of the thyroglobulin. So we've got rough endoplasmic reticulum, we've got a Golgi apparatus, and we're going to be packaging it within membrane-bound vesicles. What's different in these thyroid follicular cells is that instead of storing this within a membrane-bound vesicle, you're going to see the vesicles actually go to the apex of the cell and do exocytosis and dump this thyroglobulin into the area of the colloid, into the area of that central lumen. So we've got production of essentially the peptide component. We also need to have iodide to produce the functional thyroid hormone. And so what we're going to have is molecular pumps on the basal plasma membrane actively pumping in sodium and iodide. The important thing is the iodide is going to be brought into the cells, and it's going to be oxidized, so essentially made active by a peroxidase enzyme, and then transferred to the apex of the cell. And so we're going to take the iodide, we're going to make it active, and we're going to put it in proximity to the peptide components of the thyroid hormone, the preliminary product in the production of this thyroid hormone. Now, the important thing is that we need to take the peptide component and basically iodinate it. We've got to take that iodine that has been oxidized, that's been activated, and add the iodide onto the tyrosine residues of the thyroglobulin molecule. And so the way we're going to do that is we're going to go through and we're going to either add one iodide or two iodides. And the nice way of looking at this is if we add 1, we're going to have monoiodinated tyrosine, or MIT. Or if we have 2, we're going to have diiodotyrosine for 2 iodine residues on our tyrosine. And so we have either 1 or 2 with MIT or DIT, and then we're going to combine this on the thyroglobulin molecule so that what we have is essentially 2 tyrosine residues with iodine associated with them. And so if we've got a DIT plus an MIT, 1 with 2, 1 with 1, we get 2 plus 1 equals 3. So we get the T3 thyroid hormone molecule, or triiodothyronine. Or if we've got DIT plus DIT, 2 plus 2, so 2 tyrosine residues with combined with another 2 tyrosine residues, we get thyroxine, which is T4, indicating that we've got 4 iodine residues associated with this thyroid hormone. So we've got T3 and T4. T4, thyroxine, is about 90% of the thyroid hormone that's released, but it's much, much less potent than T3. Triiodothyronine, T3, is produced in much lower quantities, but it's much more active in regulating activities within the cells as a hormone. Okay, so we've got either T3 or T4 kind of that are stored up within the colloid, within that extracellular space. When these cells, when the thyroid follicular cells are stimulated by TSH, again, TSH from the thyrotropes from the anterior pituitary, it's going to stimulate these cells, these thyroid follicular cells, to take what's already been produced, phagocytize it, so essentially gobble it up through the process of endocytosis, bring it in. It's going to fuse with lysosomes, that intracellular digestive system that we've talked about before. We're going to bring materials from, in this case, the colloid, combine it with the enzymes from the lysosomes, put it together, and they're going to start to digest these things. But they don't digest it completely. Basically, what they're going to do is take that precursor thyroglobulin molecule and cleave off either the T3 or the T4, so that we get thyroxine or triiodothyronine, which then can be released into the area where we're going to have a capillary bed where it can pick up this hormone and transport it through the body. Now, the target effects of the thyroid hormone, either T3 or T4, is generally it's going to increase metabolic rate. And so it's going to do that by promoting cell growth. It's going to increase the heart rate. It's going to increase body temperature and have an overall effect of enhancing the majority of energy requiring cell functions within the body. So we're going to activate growth, activate development, activate metabolism, increase catecholamine effects. So basically ramp up the activities within the body. Keep in mind that T3 and T4, these thyroid hormones, are going to have a negative feedback on the pituitary gland, so regulate the thyrotropes, downregulate the production of TSH, as well as having negative feedback on the hypothalamus, where they're going to inhibit the release of thyrotropin-releasing hormone, inhibit the release of the factors from the hypothalamus that stimulate the thyrotropes. So we're going to turn off the hypothalamus in its effect of interacting with the anterior pituitary and stimulating the thyrotropes. So again, a good example of a negative feedback system to regulate this. In addition to the thyroid follicular cells, we're going to have some thyroid parafollicular cells. And these parafollicular cells are essentially outside of the thyroid follicles. They're also called C cells. And the thyroid parafollicular cells, or C cells, are going to secrete calcitonin. And so you're going to have clusters of paler staining cells, often appearing clear or very pale, or in some cases kind of white in appearance, and little clusters in between those thyroid follicles. And these thyroid parafollicular cells are going to secrete calcitonin. And basically what happens is these cells are going to respond to high levels of blood calcium. They're going to recognize the high levels of blood calcium, and they're going to secrete calcitonin. And so what calcitonin does is has an overall effect of lowering blood calcium levels. And it's going to do that by causing cells to uptake calcium, so essentially to take calcium up and store it in things like the sarcoplasmic reticulum, other locations within the body. It's going to increase calcium deposition in the bone, so promote the deposition of bone, produce, essentially stimulate the production of bone, as well as inhibit osteoclasts. Again, think about from the bone lecture series we had, osteoclasts are going to be eroding at the bone, liberating calcium, where osteoblasts, the builders, are going to be depositing bones. In this case, we're going to kind of shift the balance, so we're depositing more bone and inhibiting that bone resorption, the bone remodeling process. Overall, trying to decrease blood calcium levels to regulate it. That's going to finish up our lecture on the pineal glands and the thyroid gland. Come back for part 4 of this lecture series, and we're going to talk about the parathyroid gland and the adrenal glands. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thanks, and come back for the next lecture.